Good afternoon, everyone. I'm going to tell you about a couple of my program efforts that are aimed at improving the productivity of the drug and vaccine development pipelines. But first, I'm going to tell you something about myself because it illuminates DARPA process and culture. In 1983, I joined the University of North Carolina, Chapel Hill, as a young assistant professor of pharmacology. And for many years, I studied ion channels. And I did the things that professors normally do, like write grant proposals and serve on committees. And my entire life revolved around the laboratory. Even now, I remember the excitement I felt when Eppendorf tubes first became available in different colors. Right? I'm speaking for a lot of you, am I not? But after 15 years, I started getting bored. And I was losing my interest in science, which had been a part of me since the 6th grade. And I was even starting to wonder about life on the outside. I wanted to go to strange places. I wanted to see really cool things, and not just on TV. And even more than that, I wanted a motorcycle. So in 2001, I turned to the internet to try to figure out what to do with the rest of my life. And there I found a think tank called the Institute for Defense Analyses in Alexandria, Virginia. When they offered me a position, I accepted it without hesitation after having been in academia for 18 years. Now, to be perfectly honest, when I accepted that position, I had no idea what it was. I knew that IDA performed technical and scientific analyses for the Department of Defense, but the details of the job were really kind of fuzzy, at least to me. And even worse, a lot of the details were secret, so they couldn't tell me until after I got there. No matter. Sold our house, packed up my wife and cat, and we moved to Northern Virginia. So I started at IDA as a science advisor to a number of DARPA program managers, and that's when the culture shock hit. See, as a scientist, I'd been taught that anytime you had a grant proposal or a journal article, you analyzed it very carefully and found all the things that were wrong with it. And the more things you found that were wrong, the greater the admiration from your colleagues. But DARPA program managers seem to be doing things differently. What they seemed to be doing was looking for specific parts that were either original or innovative and pursuing those, even if they didn't know where it might lead. At other times, the program manager seemed to be looking for parts that fit into a grand or long-term plan or something that he called a vision. I had no idea what that meant at the time, no. So when I joined DARPA in 2011, I'm sorry. Now I'm right? Okay, good. So while I was undergoing this attitude, I'm sorry, let me go back to where I was, yeah. So the program manager was looking for something that fit into a vision, which of course, as I just said, I didn't understand and might not even now. But it took me at least, looking back, it took me at least 2 years to change my perspective from one that focused on what are all the things that are wrong to a perspective that says, well, what's right and what's worth pursuing? Now, while I was undergoing this attitude readjustment, DARPA was starting to take an interest in drug and vaccine development. In 2006, DARPA asked me to identify nascent technologies that might be used to improve the productivity of these pipelines, which, as you know, suffer from terrible attrition of candidate compounds at every step of the way, while consuming huge amounts of time and money. So when I joined DARPA in 2011, I knew that the pipeline was letting us down, and I wanted to do something about it. Now, one of the big bottlenecks in the pipeline is that animal testing only predicts the human response to a drug or vaccine about 10% of the time. So now that I was a DARPA program manager, I knew what a vision was. I said, what we need is a box or a platform into which you would place your drug or vaccine candidate, and out would come data. I wasn't sure what it was, but out would come data of some sort that you could use to then predict how a human would respond to the drug or vaccine. This vision was described in a public announcement that outlined the problem that we wanted to solve and asked for proposals from anyone really, but asked for proposals as to how this platform might be constructed, what was going to go inside, how long it was going to take, and what was it going to cost. And as a consequence of that open competition, we let contracts to two large teams, one team led by Don Ingber at the Wyss Harvard Institute, and another team led by Linda Griffith at MIT. Members of the Harvard Wyss team have a demonstration set up across the hall, and so later today, please visit their demonstration and take a look at their very, very innovative approach to this problem. The MIT platform is quite a bit different. The MIT platform relies on individual microorgan modules that plug into a base plate that will manage the circulation of an artificial blood supply amongst all the microorgans. In 2.5 years, when this program is over, I'm expecting both teams to demonstrate at least 10 different microorgan systems on their respective platform, and these systems have to be interacting with each other in a physiologically and pharmacologically relevant manner, and able to predict human responses using the platform before the drug or vaccine actually goes into a human. Now, the second program effort I want to tell you about came about due to the convergence of a long-term interest of mine and a relatively new one. And the relatively new interest was a publication, a report from the CDC in late 2013 describing the threat to the United States from antibiotic-resistant organisms. And as you've heard before, the news on this front is not good. We're starting to see more and more infections in the United States and throughout the world that cannot be treated by any known antibiotic. And part of the reason we've got into this fix is that microorganisms can develop resistance to a new antibiotic in only about a year. And the pipeline for producing new antibiotics, our drug development pipeline, cannot keep up, having produced only about a dozen new antibiotics in the past 15 years. Well, while we were mulling this over, my team and I had been revisiting something that I talked off and on about for several years, and that was my interest in bacterial social systems. And in particular, I was very interested in how do bacteria wage war against their competitors. As I'm sure you're aware, bacteria fight over food, over territory, and sex. So they're a lot like us. But as we looked through the bacterial warfare literature, we came across something that we had never heard of before, and that was bacterial predators. So it turns out that just as cats prey on mice and wolves prey on sheep, there are bacteria that prey on other bacteria. Who knew, right? So the particular bacteria that we're most interested in at the moment is called Bacterial Vibrio bacteriovorus. And what it does is it swims around propelled by this flagellum, and when it finds a gram-negative prey bacterium, almost any gram-negative bacterium will do, it adheres, it attaches to part of it, and then it enzymatically drills a hole into the prey bacterium. And when the hole is made, it then wriggles itself in, it feeds on the contents, it reproduces, and then in doing so, kills the prey. Now you can see this entire life cycle in this video from Liz Sockett's lab in the UK. And what you're seeing is, if you look over here, you'll see a predator, and it's attached to this oblong object, which is the prey. And over here is a prey that already has, in the lower left, a predator in it. And you saw them burst out as they completed their life cycle. When the video restarts, what you're going to see is coming in from the lower left will be a, there he goes, a predator races in, reaches into that, breaches the wall, and enters that prey. Now, right now you're thinking, where can I get one, right? And it turns out they're ubiquitous. You can find them, they're in soil, they're in water, they're in you right now in your intestinal microbiome. Now, why was this so exciting to me as a DARPA program manager? The reason this was so exciting was that Daniel Kadori at Rutgers had published in 2013 a study showing that these particular predators readily prey on antibiotic-resistant pathogens that had been isolated from humans. So right now, we are just starting up a program called Pathogen Predators, and this program is going to test the feasibility of using living bacterial predators as a therapeutic against antibiotic-resistant infections. So to finish up, what I'm going to do is I'm going to speculate a little bit about the future I'm helping to build. We talked about what we expect from the microphysiological systems program, and what we expect are profound changes in the drug and vaccine development pipelines as they currently exist. But think about the impact that this platform might have, or probably will have, on the overall biomedical and biological research enterprises. We just talked about the pathogen predators. If these can be used as an effective therapeutic against antibiotic-resistant infections, we will be opening up an entirely new line of research and approach to the treatment of antibiotic-resistant infections. You know, we seem to know so little about the world of microorganisms around us. Who knows what else is out there that we might use for our own benefit? Finally, I have a little crib here to remind me to tell you about a program called Rapid Threat Assessment. This is a program that is developing tools that will determine the complete molecular mechanism for any drug or biologic as a function of time, and do that in 30 days. Now imagine what we can do with that information. We'll finally be able to create computer models that will predict what any drug would do to a cell before the drug is even made. Or from a disease standpoint, such a model would tell us which specific biomolecules we should be targeting so as to effect a treatment or perhaps even a cure. Tomorrow, William Old will be speaking. Will is a team leader from the University of Colorado Boulder team, and he will be talking about his adventures as team leader under this new program, and I really urge you to hear him tomorrow. Finally, you've heard us numerous times say, you know, we need your ideas, and it's really true that we need new ideas. Anything that will lead to a new capability or a significant advance in biological science, or even better, a major upheaval in biological science, we are interested. So I urge you, if you have ideas, contact us, come see us, work with us, because we cannot do this alone. Thank you very much.
All right, well, it's time for another break, and let's reconvene at 3:30. Thank you.